All right, so on to chapters 26 and 27. So starting off with 26, which is all about industrial microbiology. I'll go into details about all of the industrial applications of microbes. So this is everything from today's version of preserving food to biotechnological applications in the case of genetically engineering or manipulating yeast or bacteria to produce all kinds of different products. So historically, as far as the method for food preservation, it was via drying or desiccation using a high concentration of salt or sugar to preserve food, which is still done with jams, jellies, also salted meats, for example. And then also fermentation. So the products of fermentative metabolic pathways like lactic acid or ethanol to preserve a food or a beverage. So nowadays, as far as our method for preserving canned foods, canned goods must undergo this specialized cycle of autoclaving or cycling of steam autoclaving to ensure that the food that us as a consumer will be ingesting after we purchase this can at the grocery store is free of Clostridium botulinum spores. So these are the spores that are notoriously causing botulism or cases of that food poisoning. And so using this moist heat method of autoclaving, so heating up the food at a very high temperature using this moist heat or steam, and multiple cycles of this seems to do the job of destroying those spores. So these are actually the commercial canning machines here. So this is kind of a huge autoclave machine that again has high pressure, high heat, moist heat that will destroy these endospores. So there's a specialized treatment here, the 12D treatment cycle. Now, as far as preserving food in a package, let's say chips, right, that you purchase at the grocery store, the contents of that package must undergo this aseptic packaging protocol. So everything within the contents of the food and also the package itself needs to be sterilized and aseptically filled. So as far as the methods for ensuring that those packages are free of any microbial contamination, again, I showed you the autoclaving, which would be for canned goods, but for packaging, a lot of times ionizing radiation can be used, and we know of the appropriate doses that are necessary to destroy various organisms. So if you take a look at the human body, 0.005, this is grays or kilogray, this is a unit for radiation. So 0.1 here, much lower than, check out the amount that is needed to kill a virus, so 10 to 200 kilograys. So we know the specific saturation point or threshold for each one of these microbes, and so we can then tailor the ionizing radiation treatment to destroy all of the potential pathogens that may be in that package. Also, high energy electrons or pressure can be used as well in the food preservation process. Now, since we're on the topic of industrial microbes, I'm going to mention a little bit about some of the foodstuffs or beverages that are classically used or produced via the natural metabolic products of some microbes. So cheese, for anyone who loves cheese, and I'll talk about a pairing with cheese that's also dependent upon microbial processes, which would be wine. But cheese is the result of microbial metabolic pathways that will basically convert milk into the solid and the liquid form. And it's typically bacteria, lactic acid bacteria, that will carry out this process. So the curd is the solid portion that ends up being converted to any type of cheese dependent upon the bacteria, and also rennin is part of that solid portion. So casein, this is where all the protein is derived from. And then the whey is the liquid. So this is actually separated out and is not consumed when we eat cheese. And typically the hard cheeses are produced by lactic acid bacteria, whereas the semi-soft cheeses can be additionally ripened by a fungus, so Penicillium on the surface. And so this is just a few images at the various stages here, so separating out the curds from the whey here. And then the curd is milled to allow for even more drainage of the whey and then compressed into blocks for ripening. The longer that the ripening period lasts, the more acidic or sharper the cheese will be. Now, also a good pairing with cheese is wine, which is also dependent upon microbial processes. So this is dependent typically on yeast, so Saccharomyces cerevisiae or Eubayanus, also used to make beer. So you start off with grapes, and it's actually the grape juice that gets fermented to wine. So the grapes are tested and picked, crushed, destemmed, and then they are added to the fermentation tank, which is sterilized. So sulfites are added to kill undesirable yeast and bacteria. The yeast inoculum is added, and the result is pressed to separate the solids from the wine. The wine is then clarified into settling vats, filtered, aged, and then bottled. And so depending upon the type of wine that you have is usually dependent upon the specific grape that is used. So they can vary in acidity and flavor in the end. Also, the yeast that's used, sometimes there is a co-inoculation of different types of yeast that can affect the flavor in the end. But the biggest thing with making beer and wine is to make sure that you don't have any contamination. So with beer, yeast is also used. So this is fermented starch, and in the malting process, the germinating barley converts starch to maltose and glucose. For sake, this is fermented rice. So this is rice wine. Rice starch is converted to sugar by a fungus, Aspergillus. The wine is fermented plant sugars, and in the process, for both wine and beer, yeast takes the sugars and converts it to ethanol and CO2. And grape wine also can require, I mentioned before, co-inoculation with different types of yeast, sometimes malolactic fermentation, which can be a bacterial derivative. And sometimes what happens here is that you can have contamination of the wine with different strains of bacteria that will take the ethanol and convert it to acetic acid. So this is not good. This is when the wine can taste a little funky or turns a different color from it being left out too long or not properly capped. So that bacteria will enter the wine and then convert it to something that tastes like vinegar. So these are kind of just 3 different groups of microbes here that are involved in metabolic pathways in making food or beverages. So sugar, the yeast will convert that sugar to ethanol and carbon dioxide, malic acid, lactic acid bacteria to lactic acid, and then the contaminating bacteria will take ethanol and convert it to acetic acid. So now there's also this whole new wave of technology that has come out as a result of our knowledge of how DNA is read and how we can manipulate that DNA. And microbes are a very good source for this because we can culture as many of them as we want in a lab. We can genetically manipulate them. We know their genome sequence, so we can easily add pieces of DNA into their background or take out those pieces of DNA and manipulate that organism to make whatever we want. So this is what we call a biotechnological application. So this would be using microbes, cells, or cell components to make a product. And so I'm just going to go over some basics here if we're talking about using bacteria. So if we start off with a bacterial cell and we want to exploit that bacterial cell, change its genome, so add a specific DNA sequence into the genome of the bacteria to encourage it to make a specific product. So let's say that we wanted the bacterium to crank out a lot of insulin. We could actually take a gene sequence that coded for that product, and we could clone it into a bacterial cell, and we would clone it into the bacterial plasmid. So if we wanted to clone a piece of DNA into a bacterial cell, we need three things. We need a way to cut the native chromosome, cut the native piece of DNA that's already present in that bacterial cell at specific sites. We also need a carrier molecule to hold DNA for cloning, and we need a host cell where the DNA can be copied. So in order to cut that piece of DNA, we will use this enzyme called a restriction enzyme, and this is basically just an enzyme that kind of acts as molecular scissors. So it basically is designed to target a specific gene sequence, so AATCCG. But what's unique about these enzymes is that they will cut a gene sequence that looks like a palindrome. So a palindrome actually reads the same way forward as it does in the reverse. So I'm going to show you guys some examples of that in a minute. But just think of it as molecular scissors. So this is an example of one. I said it's a palindromic sequence, right? So it's read the same way forward as it is backwards. So GAATTC, backwards GAATTC. And so if we wanted to actually cut out a piece of DNA, we would use a restriction enzyme. So the restriction enzyme EcoR1 actually recognizes this unique sequence, and it will cut it in such a way that it will leave these sticky ends. So the matching sticky ends of different pieces of DNA can be annealed together, and they are annealed via this enzyme called DNA ligase. So this is, again, cutting a piece of DNA and adding new DNA into that stretch of a sequence. So this is actually indeed one of the ways in which you can clone a piece of DNA. And there's all different restriction enzymes that are unique according to the sequence that they recognize. So EcoR1 recognizes GAATTC. HIND3 recognizes AAGCTT. BAMH1, GGATCC. Again, remember, they're unique because they recognize palindromic sequences. They're stretches of DNA code that read the same left to right as right to left. And so if we would like to clone a piece of DNA, I mentioned that you need a carrier, right? You need a host cell, which would be the bacteria, and you also need a piece of DNA that can carry that cloned gene. So we have the native chromosome that's present in every bacterial cell. Every bacterial cell requires that piece of DNA to thrive. But then we also have these exogenous, these extra pieces of DNA called plasmids that naturally bacteria can swap in an effort to gain a selective advantage. So they could swap plasmids to gain antibiotic resistance, for example. But we also can manipulate bacteria to pick up these pieces of DNA in the case of biotechnological applications. So we could actually take a plasmid and work with it in a test tube to actually cut in or clone in a specific gene and then reintroduce this plasmid back into the bacterial cell. So here's our bacterial cell. Here's the native chromosome that cell needs to thrive. So it requires it. It's an essential piece of DNA. But we could also manipulate a completely different piece of DNA, this plasmid, reintroduce it, and the bacterial cell will actually treat that plasmid like it's its own native chromosome. And the way in which we can actually select for cells to keep that plasmid is you can put a selective marker on it. So for example, we could actually put an ampicillin resistance gene onto this plasmid, and we could grow bacterial cells on plates that have ampicillin. So the goal again here is to clone a specific piece of DNA into a plasmid and cut that piece of DNA using the restriction enzymes. And again, like I mentioned, the plasmid typically will have a selective marker that then you can introduce into this bacterial cell and select on ampicillin plate. So I'm going to come back to that in a few. But typically the most common host cell that we'll use is E. coli because it's very easy to culture in the lab. And after we introduce that cloned piece of DNA into E. coli, it can be recovered from the host cells and purified for further use. So that insulin gene that I mentioned before, we could introduce into this plasmid at a specific restriction enzyme site, introduce the plasmid into the bacterial cell, grow the bacterial cells, and what happens is that the genes on the plasmid will be expressed, so insulin will be expressed, and we can purify it. So how do we actually amplify or produce that insulin gene so that we can insert it into the plasmid? So we would do it via this process called PCR. So PCR stands for polymerase chain reaction. This is a method for amplifying DNA using specific cycles of denaturation, annealing, and DNA polymerase-directed DNA synthesis. So basically all of this is just a fancy description for amplifying a specific gene sequence up to about here 68 billion copies. And once we have this wanted gene amplified, we can then clone it into that plasmid. So I'm just going to briefly go over how PCR works. So the first step of the process here involves heating up, and essentially the PCR machine will heat up the DNA so that it will separate. So you have this template DNA that then will separate into two strands, and that will be used for a template for a future cycle of replication. So number one, the DNA is heated to break the bonds, and the two single-stranded DNA molecules serve as templates. Then those templates serve as the way in which the DNA can be amplified for future generations. So you have one single strand of DNA that then is used as a template for the primers. So there's a forward primer and a reverse primer. These will anneal to that specific DNA sequence. So I'm going over this just in text here. I'm going to show you guys in a minute a visual of how this occurs. So you have these short nucleotide sequences. They're used as primers for DNA replication. They're mixed with the DNA, and also they bind to complementary regions on the single-stranded DNA. This takes place at a lower temperature. Those primers are generally 20 to 30 nucleotides long, and they are produced or synthesized in the laboratory. Then after the primers anneal, then you have a Taq polymerase that's added and will start to synthesize the new strand. So these three steps make up one PCR cycle. Once these two strands have served as templates to make two new DNA strands, then the cycle starts over all again, and you get those two strands separating, and there's 4 templates rather than the original 2. So it's an exponential cycle here. So again, so just for a visual, so you start off with your template. So if this is the stretch of DNA that we want to amplify, so this would be an insulin gene. What happens here, you heat it up, it unwinds the DNA double helix, and you mix this with these primers that I mentioned. So there's these short stretches of DNA that will complementary base pair with the sequences. So once the mixture is cooled, lowering the temperature promotes base pairing between the primers, and the DNA strands start to be copied. So they're going to hop on here, and we need the polymerase to recognize the primers and synthesize the new strand. So we started off with, again, one DNA double helix, 2 strands. It unwound. We have 2 new templates, 2 new DNA double helixes. Now, these will actually undergo another cycle of the PCR reaction, and they are going to unwind from heating. They are also going to serve as templates for the next cycle. So primers hop on, they complementary base pair, the polymerase synthesizes. Now we have 4 new strands. And repeating the sequence of reactions will double the number of DNA fragments each time. So you end up with billions of fragments that are rapidly synthesized. And if you check it out at cycle number 30, look at the number of copies that we have here. So usually this goes up to around 35 cycles or around 68 billion copies of that one piece of DNA that is synthesized. And once you actually do this and you clone that stretch of DNA into a plasmid and introduce it into the bacterial cells, then remember I said that plasmid has a selective marker. So we can actually plate those cells on an ampicillin, an agar plate containing ampicillin, and only the cells that have taken up that plasmid that has that resistance gene will grow. So there's methods or strategies that will definitively tell you whether these cells have actually picked up that piece of DNA. I won't go into that, but just as kind of the general picture here, just know that following this whole cloning process, you would actually grow the cells and screen for positive clones using that selective marker, right? So we can use these biotechnological applications to produce almost any type of industrial product that we want that could be synthesized using small microbes like bacteria and yeast. So everything from different enzymes to antibiotics, steroids, even non-microbial compounds like insulin that aren't generally produced. As long as the cell can tolerate those compounds and they use the same molecular language, the sky is the limit as far as what we can actually produce. And there's this whole new wave of synthetic biology which actually creates a cell from scratch. So it's a man-made chromosome that then we can genetically manipulate to make anything that we want. And also, even now, there are companies that are attempting to synthesize alternative sources of energy, or on a mass scale, taking all the biomass and converting it to methane or ethanol that could be used as a substitute for petroleum, for example. So this whole Go Green movement, really at the forefront of this is the potential in microbes to produce these alternative sources of energy. And then transitioning now into chapter 27. So that was a little bit about industrial micro, and now we are going to focus on the environmental microbiology. So microbes are quite diverse. They can live in almost any given environment. So everything from a nuclear reactor to a hot spring to a salt lake. They use a variety of carbon and energy sources. They can grow under many different physical conditions, and especially the archaic microbes. Those are called archaea because they're the arcane, or the old ones that have been around since the beginning of time. And all of the archaic microbes always live under these extreme environments, so extremes of pH, temperature, and salinity. And all over the place, we see microbes being involved in different types of symbiotic relationships. So there's a few listed here, but again, as a review, symbiosis is referring to two differing organisms living together in a close association in which one benefits. So at least one benefits. So mutualism would be both, commensalism would be one and the other is unharmed, and parasitism is one is harmed and the other benefits. But just two examples here of how microbes and animal cells can work on this symbiotic level. Ruminants, for example, multiple stomachs that are found in sheep and cows, they contain these unique bacteria that are able to metabolize cellulose. And so when these animals consume plant material, they can degrade the plant material appropriately and use it as sources of energy. Also, mycorrhizae, these are a specific type of a fungus that grows on plant roots, and this actually helps to extend the surface area of the root. So it aids in the plant's ability to extract water from the environment and also nutrients in the surrounding area. And there's commercial uses of this. So just looking at these seedlings, the one on the left has been inoculated with the mycorrhizae, the one on the right has not, and you can see just the difference here. The relative growth, the seedling on the left has that inoculation. They grow better, so this can be used as a commercial application. And our bodies, the human body, is a great example of symbiosis. So we have billions of bacteria that cover every inch of our bodies. Also, the main location is within the gut, and there's this whole gut to brain axis that we have seen over the years. New research is suggesting that there is this crosstalk between the chemicals that are produced in the gut by the normal microbes and how that can affect brain function and vice versa. So the ability of the brain to influence intestinal microbiota. We see microbes produce a large majority of neurotransmitters in the gut environment. There's also a whole other brain that exists. There's neurons that actually line the gut. And so this is actually a really hot topic that's going on now, is we're finding links even with autism or mental illness, how the brain is affected by these normal microbes that are growing in our gut. And if you look at all of the different types here, so they're in our stomach, they're in the large and the small intestine, the large intestine or the large colon being the major site. And again, as far as the benefits here, so it's not only how the brain develops or functions, but it also aids in digestion. They produce the majority of IgA antibodies, so immunoglobulin A antibodies. They also produce vitamins. They metabolize carcinogens. They kind of serve as our first line of defense for any pathogens that enter the digestive tract. So the benefits go on and on. That list is a very exhaustive one. Also, microbes are very important for contributing and maintaining the majority of biogeochemical cycles. So these are all of the cycles involving specific elements in the atmosphere. So nitrogen, carbon, sulfur, phosphorus. And when I say recycling, this is referring to basically the oxidative and the reductive processes. So this would be gaining or losing electrons throughout the cycle. So the carbon cycle is basically balanced by changes in fixation equaling changes in respiration. And this is maintained by a combination of respiratory processes from animals and plants. So photosynthesis is a huge part of this. Also decomposition from fungal and bacterial microbes. Also, the ability of, and this is where this is really important for the carbon cycle. So for each one of these cycles, I would like you guys to kind of focus in on where the microbes are really playing a significant role. And in this cycle, it would be in the aquatic environment. So aquatic bacteria and also algae and cyanobacteria, big one here. This group plays the most important role in converting carbon dioxide to oxygen. They actually are #1 on the list for this, and so it's thought that actually part of the whole global warming issue has to do with changes in the populations of the algae and the cyanobacteria that are carrying out this process. So also the nitrogen cycle. I would like the class to be aware of these 4 metabolic steps here: ammonification, fixation, nitrification, and denitrification. So if you look at the cycle, again, it's a balance of the oxidative and the reductive processes of taking free nitrogen gas, for example, and converting it to fixed forms. So these microbes actually continue the cycle. Most importantly, so ammonification here would be fungi will take the nitrogen and convert it to ammonia. Also for fixation, this is carried out by plants again, so this is taking nitrogen, converting it to ammonia, more of a fixed form from the free nitrogen gas. For nitrification and denitrification, this is carried out by bacteria. Nitrosomonas will take ammonia and convert it to nitrites. We also have denitrification that is carried out by similar bacteria here. Nitrobacter converts this previous step from nitrites to nitrates and then back to nitrites. So the denitrifying bacteria, I would like you guys to remember the names. Pseudomonas is a big one, Bacillus, and Paracoccus. There are others, but these are the major groups. So this is all of the ones that I just mentioned here. So good to know the names: ammonification, fixation, nitrification, and denitrification. And then we have the sulfur cycle, which is again involved in microbial processes are involved in recycling different elemental forms of sulfur. So Thiobacillus will take the elemental sulfur and convert it to sulfate. Also, we have reductive processes here that will convert it to hydrogen sulfide. And the 2 major groups of genera of bacteria that are involved in this process are Desulfovibrio and Thiobacillus. So this is, again, the sulfur cycle. These are the ones that I just mentioned. And also, since we're on the topic of environmental micro, I'll mention a little bit about kind of some of the varieties. So microbes that will function in an environment where there's no light. They would serve as primary producers. There are endolithic communities, also bacteria that live in rock. So if anyone has heard of the theory panspermia, this theory is an idea that the first forms of life on Earth were the result of an inoculation from a rock from outer space. And so this idea of a meteorite or this astral rock that actually got introduced to this planet accidentally. There's some scientific basis for this, but the idea here is that there are bacterial populations that can survive living within rock, and they are chemoautotrophic bacteria. So they can actually take insoluble phosphate and convert it to soluble forms. Even Thiobacillus, which is found in bird guano or bird poop, can take the soluble form and convert it to insoluble. And we know phosphate and phosphorus is a critical element that's found in DNA. So it's a very important molecule that makes the deoxyribonucleic acid foundation and also ribonucleic acid. So both of those components require phosphate. And then in the environment, there are some compounds that are highly resistant to degradation. So this would be considered a xenobiotic. Naturally, organic matter is easily degraded, but some are more highly resistant. So all of these on the right here, certain antibiotics will build up in the environment, carcinogens, alcohols, petroleum products. And so they're more resistant, but we do have microbes that we can genetically manipulate or encourage to break down some of these resistant compounds. So bioremediation is an example of this, where we can use Pseudomonas bacteria to break down hydrocarbons and some of the toxic compounds that you find in oil, like toluene, naphthalene, benzene. Those can be used as targets for microbes. So they'll basically take those toxins and convert them to non-toxic compounds. So this is bioremediation. Also, the bacteria, the growth of that bacteria can be enhanced by the addition of fertilizer, nitrogen and phosphorus fertilizer. Also, bioaugmentation. This is similar to bioremediation. And then composting. This is a natural way of arranging organic waste to promote microbial degradation, and this would be by thermophiles, so the bacteria that actually like heat. This is just showing you pictures of natural decomposition in the case of compost or solid municipal waste. And then transitioning into a little bit about the aquatic environment. So I'll talk about both the freshwater and also saltwater in the case of an ocean and the types of microbes that you would find there. Okay, so as far as the particular anatomical zones of a freshwater aquatic environment, there's the littoral zone, which is along the shore. This is where you'll find the plants, the photosynthetic organisms. Also along the limnetic zone, which is the surface of the open water along the shore. And here you'll find more algae, also the cyanobacteria, which are the major producers of oxygen. Again, they'll fix carbon dioxide to oxygen. And also the profundal zone, so this is deeper water under the limnetic zone. And here you'll find anaerobic purple and green photosynthetic bacteria. And then the benthic zone is deeper in this environment around the bottom sediment where there's very low light, low oxygen. And you would find organisms like Disulfo vibrio, methanogens, and also Clostridium living in this zone. Along the saltwater or seawater environment, similar terms, littoral, sublittoral, and more algae that you would find here, especially along the littoral zone, green algae, euglenoids also. Then in the sublittoral zone, the unicellular green algae, diatoms, dinoflagellates as well. And there's also a benthic zone, which would be more the bottom sediment. In the top 100 meters of a seawater environment, this is where you'd find the phytoplankton. So these guys, they're photosynthetic cyanobacteria, and they are the major carbon fixers. So they're taking the carbon dioxide, Prochlorococcus, Synechococcus, they're fixing it. They're also fixing nitrogen, Trichodesmium, and then also can be decomposed by Pelagibacter ubique. So this is just another bacterial species that you would find within this zone. And also you would find below 100 meters bioluminescent, potentially, bacteria that can produce a photon in light, or to produce light from photons. And this would, so bacteria that produce luciferase, this is an enzyme that can convert FMNH2 to FMN into photons or release light. Some of these bacteria are associated with organs around the eyes of fish, so it actually allows them to see more appropriately in this environment. And we can exploit the fact that we know there's a specific gene that codes for this enzyme. We can actually snip out that gene sequence and clone it into bacteria and use this as a genetic tool to track specific changes that have occurred. And also we can use this enzyme as well for other biotechnological applications with immunofluorescent imaging and diagnostics as well. So a little bit about how water is treated for, in the case of municipal wastewater, and also for sewage treatment. So both of these would be in an effort to prevent contamination in drinking water. So all of the water that comes out of your tap has undergone specific cycles of sterilization and also testing to ensure that it is not contaminated. And so these are actually pictures of diatoms that may produce neurotoxins when associated with shellfish, and you consume that seafood can actually result in food poisoning. And also in the water environment, mercury can build up in larger animals. So typically the largest fish has the highest amount of mercury, and this ends up being concentrated in the human body. We have no real way to break it down, so it just ends up accumulating, and it has been shown to cause neurological effects. So for anyone who likes sushi, eating the biggest fish is the most hazardous. So that would be tuna, for example. Also, there's this case in an aquatic environment of eutrophication. So this would be an overabundance of nutrients, and this is caused by a buildup of organic or inorganic matter, so a lot of phosphates, nitrogen, which can cause the overgrowth of algae, so these algal blooms. And this is where you can have an overgrowth of the diatoms that can produce those neurotoxins that I mentioned before. Now, in testing water to ensure that it's free of contamination, the number one group of microbes that they screen for would be the coliforms. So these are the lactose fermenting bacteria that are associated with fecal contamination, and also Enterococcus they screen for as well. And they're looking for the NPN, so the most probable number out of 100 mils. And again, coliforms is at the top of the list. So these are aerobic or facultatively anaerobic, gram-negative. They don't form spores, and they're able to ferment lactose to acid plus gas within 48 hours. So they're looking for the NPN out of 100 mils of water. And there's different ways to screen for it. There's an enzymatic test. There's also a filtering method here, so just to see how many colonies grow. Ideally, you don't want any risk for fecal contamination. You could screen for it in the case of E. coli, which would be the number one bacteria associated with fecal contamination, by adding this MUG compound. And so if E. coli is present, you'll have beta-galactosidase in the water sample. When you add this MUG, this enzyme will convert MUG to a fluorescent compound. So it will actually fluoresce in the jar that you're testing for coliforms. So very, very important that the water is free of any fecal contamination. And so there's this whole process of municipal water purification treatment that occurs here, where you have this initial step. The raw water is held in a reservoir and the particulates settle. So the supernatant, or the soup, is then pumped to a mixing tank that's mixed with a bacterial flocculant. And that flocculant will actually carry out oxidative processes, which will break down the organic matter. It then is settled once again. The soup is pumped into a particulate filter where the water is then filtered. Any residual particulates are filtered out, and it's sent to a storage tank, and usually chlorine is added, ozone, or exposure to UV light to ensure that when we consume that water, that it is free of any bacterial or pathogenic contamination. Also, the whole process of wastewater treatment, I'll talk about a septic tank and then municipal water treatment. So in a traditional septic tank here, you have this sewer line that leads to the septic tank, and then leaching fields that will act to essentially disperse the contents from the septic tank. And it's all of the bacteria that's present in the oil, in the soil, that will break down that organic matter. So it kind of acts as similar to composting. It's the oxidative processes that break down the organic matter. There's also oxidation ponds. So for small communities, You have one pond that will settle the solids and then pump the water to the second pond where you have bacterial composition of dissolved organic matter in that water environment. And then lastly, we have municipal sewage treatment, which contains 3 different phases. So please remember what happens in each phase. So in the primary treatment phase, you have the solids that settle out in a cycle of disinfection. Secondary stage, you remove much of the biooxygen demand, so much of the organic matter. Also disinfect here, and the water can be used for irrigation. And then lastly, this tertiary phase where you're removing any of the remainder of the BOD, the nitrogen and phosphate, followed by an additional cycle of adding chlorine or disinfection, and then the water is drinkable. So you basically start off with sewage and end with potable or drinkable water. And so this is the cartoon image of it. So sewage is sent to a sedimentation tank where the solids settle out and are sent to a landfill. The supernatant, or the liquid, is then sent to this activated sludge system and also a trickling filter. So it will undergo aeration and microbes will oxidize the organic matter. And then you have this last stage where it undergoes disinfection, chlorination, and you end with drinking water. So I would like you guys to remember that it's really the secondary treatment phase where the majority of the microbial processes occur. So the biological oxidation occurs. And this is just showing you a picture of that aeration tank in the secondary phase. This is frothing from aeration, so this is the microbial oxidation step. And also this trickling filter system is another portion of the secondary phase. So usually it consists of this rotating spray arm for the incoming sewage, so the liquid portion following the settling phase, and it's covered by a rock bed or a plastic honeycomb. The effluent is shipped off to undergo further oxidation and then chlorination and disinfection. So along with the aerobic sludge system and the trickling filter, also in the sludge digester, there's an anaerobic phase here where you have, again, it's microbial processes, but it's lacking oxygen. And usually this is attached to a gas outlet because of all the methane gas that is produced. But this is part of the secondary phase before the sludge is actually shipped out to the landfill. And in that secondary phase, you still have 50% of the original nitrogen and 70% of the original phosphorus that's left over. The tertiary treatment removes these by filtering through sand and also activated charcoal and chemical precipitation as well. So that is the end of chapter 27, and I will see you guys for the viruses.